Thank you very much for the opportunity to speak today. I'm Yohei, and I'm going to talk about genome sequencing in bacteria. So I'm going to use the term genome sequencing, whole genome sequencing, and high throughput sequencing interchangeably for the purpose of this talk. So use of high throughput sequencing in clinical microbiology, I think, has two broad applications. One is infection control and hospital epidemiology, where it can be used to detect and monitor pathogen transmission in hospital as well as the community, and sometimes identify reservoir exposure and route of infection. The other major application is clinical diagnostics, and this includes pathogen identification and prediction of antimicrobial susceptibility. Broadly speaking, there are two kind of groups of technologies. One is a short read sequencing, and the other one is a long read sequencing. And short read sequencing is the most commonly used one and is able to identify gene presence, absence, single nucleotide variants, as well as small indels, insertions, and deletions. It's good for species identification, probing of mutations, identifications of resistance and virulence genes, strain typing, and phylogenetic analyses. At this point, it's readily accessible and has also become relatively inexpensive. But it still takes some time to run, and so it tends to be processed in batches, leading to delays in the data output. Long-read sequencing, on the other hand, is able to resolve genomic structures, identify rearrangements, and also gene duplications. And these are difficult to do with short-read sequencing. So long-read sequencing is good for obtaining closed genomes, and investigating horizontal gene transfer, including elements like plasmids, phages, prophages. At least the Oxford nanopore technology has the capability to put out data in real time. But cons include the price of sequencing, at least PacBio sequencing, is relatively expensive. And Oxford nanopore technology has higher error rates than typical short read sequencing. So with that in mind, we'll look at some applications of genome sequencing in bacteria. Now, Dr. Rosen may be going in more detail about sequencing for infection control purposes. So I'll highlight an example here where this is in 2013 when we worked on analyzing an Acinetobacter outbreak. We're using pulsed field electrophoresis, PFGE, at the time. But when we were examining another outbreak, this one with Serratia in 2020, we're no longer using PFGE. Instead, we were using single nucleotide variations detected by long-read sequencing. So things have really changed even in the past few years in this area. And sequencing-based epidemiology complements clinical epidemiology, and it provides a much higher resolution compared with the conventional methods like a PFGE. And another big advantage is that the data are fully digitalized and shareable. There are some outstanding challenges, though. One is the need to customize analyses based on goals and species, and so on. Turnaround time is not quite yet ideal. And from public health and patient care response perspective, throughput and speed tends to be more important than complexity of analysis. And that kind of the way we approach sequencing and analysis may be a little bit different from when we're doing it solely for research purposes. Many studies have reported successful correlation between resistance predicted by high-throughput sequencing and actual phenotype, resistance phenotype of the bacterial isolates. And we know that this works better, especially in species that have stable genome structures, like Mycobacterium tuberculosis or Staphylococcus aureus. And also when one-to-one correlation between gene presence absence or specific mutations and phenotype exist. Here's an example of work around prediction of AMR. This was done by our previous ID fellow, Dr. Babikar. So his question was, can we use just web-based online tools to predict AMR? And so using MRSA and VRE genomes, he was able to show that over 99% concordance between resistance predicted by high-throughput sequencing data using readily available tools like ResFinder and CARD, and actual phenotype of the bacterial isolates. So you don't have to be a programmer to do these analyses. But there are instances when prediction is difficult. One is when multiple mutations impact a specific phenotype, and this happens in mechanisms like beta-lactamases. Also, gene expression can contribute to the levels of resistance, for example, with the efflux pump. And we're recently finding out that copy numbers of a given gene can also impact levels of resistance, like with ribosomal RNA genes. And these features are difficult to predict solely based on sequencing of the DNA. And the potential solutions include, one is to leverage machine learning, and the other is to integrate expression analysis. So transcriptomics or RNA sequencing. So I'll highlight an example of machine learning to predict AMR. So this is trying to predict phenotype without a priori information. So without reference database. This approach requires a lot of genomes, but it does provide an unbiased analysis, and also may be able to discover new resistance mechanisms that are not yet curated or in the reference databases. So here's an example where a group of investigators looked at a short-read sequencing data of 5,000 non-typhoidal salmonella isolates, which had been sequenced by Illumina and for which the data were publicly available. And these genome information was also accompanied by MIC data of 15 antibiotics obtained by microdilution method, also publicly available. So the authors took advantage of these data sets and used a gradient boosted model to define the ability of machine learning based prediction of AMR. So here's an example. They used a k-mer based approach. So if you look at the top row, you see that there's a short stretch of sequence, which is a k-mer sequence, and it corresponds to a part of the 10 beta-lactamase gene, and it has a very tight correlation with resistance to ampicillin, which makes sense. But these analyses are possible without the prior knowledge that TEM beta-lactamase confers ampicillin resistance. So they used 4,500 genomes to build a model that provided an overall accuracy of 90% to predict the actual MICs within a 1-2-fold dilution step. And one fortunate thing in this case was that the underlying database of 5,000 plus strains was unbiased as well, included susceptible and resistant isolates. So that gave an opportunity for a comprehensive analysis across the spectrum of MICs, which the authors discuss led to accurate prediction of MICs. The next topic is integration of transcriptomic data. So Pseudomonas aeruginosa is an important AMR pathogen that resistance tends to reflect gene expression. So here, the researchers took genome and transcriptome data of just over 400 Pseudomonas aeruginosa clinical isolates and combined the data for biomarker identification. So using information from gene expression, gene presence absence, and single nucleotide variations. And the target antibiotics were tobramycin, ceftazidime, ciprofloxacin, meropenem, which are commonly used antibiotics against infections caused by Pseudomonas. And the combined use of gene presence absence, SNPs, and information on gene expression resulted in high or even very high sensitivity and predictive values. So here, addition of gene expression information facilitated highly sensitive and specific resistance prediction in Pseudomonas. So transcriptomic data are much harder to obtain than whole genome sequencing data, but this might be the future, especially for species like Pseudomonas. So we talked about turnaround time. And so the next question is, can real-time nanopore sequencing streams be leveraged to improve or shorten turnaround time of resistance prediction? So this is work done by Dr. Kamma's group at Johns Hopkins, where they took 40 Klebsiella pneumoniae clinical isolates with various resistance patterns and compared 3 approaches to sequencing against a susceptibility testing results. So one was real-time nanopore analysis. Second was assembly-based nanopore analysis. And the last one was hybrid assembly of Illumina and nanopore analysis. And in short, they were able to demonstrate an overall agreement of up to 77% based solely on real-time analysis. So these are sequence reads that are obtained as sequencing is being done. And here's the timeline of the 3 methods. And the real-time approach and assembly approach could shorten the median time to effective antibiotic therapy, hypothetically, by 20 hours and 26 hours, respectively, compared with standard AST susceptibility testing. So this is all exciting. There are still challenges before we can fully incorporate bacterial sequencing in especially clinical microbiology, including indication, which conditions, cases should be the target, workflow, costs, human resources, bioinformatics, turnaround time, data sharing, and QC. But I'd like to focus a little bit on bioinformatics, because that seems to be a challenge across the board. And this is a piece of work that we recently published called ORF-based binarized structure network analysis of plasmids. So this is for plasmids, but it can be expanded to whole genome sequencing. The idea is that we express plasmid structures as binary sequences based on gene presence and absence. So if a plasmid sequence has a given gene, it gets 1. And if it doesn't, it gets 0. So every plasmid is expressed as a series of 1s and 0s, irrespective of the direction or order of genes, which tend to be unstable in genetic structures like plasmids. And the phylogeny of plasmids are then visualized as a neighbor network. So the advantage of this approach is that it's totally digitalized and does not require the presence or common sequences among the plasmids, or by extension, genomes. Here's an example of an output of plasmid network. So this approach seems to be useful in overcoming the limitations of existing methods, including PMLSD and the direct head-to-head plasmid comparison, which could be really labor intensive. And this can be applied to plasmids of the same group, even when nucleotide sequences are not shared among plasmids of interest. So this is just one example, but there's likely to be a lot of different ways we can approach bioinformatics of high-throughput sequencing data in bacteria. Now, this is something a little bit different, and I'm going to talk about discovery from publicly available sequence data. So there's exponential amounts of sequencing data that are being deposited to public databases every day. And most sequences remain uncurated, and that presents an opportunity for discoveries. So here's a recent work on a gene called NPMB. So 16S ribosomal RNA methyltransferase is a group of enzymes that confer high-level resistance to aminoglycosides, and they do so by targeting or methylating specific nucleotides in the 16S ribosomal RNA. And only one such enzyme, called NPM, was known to modify position 1408. And this discovery was made almost 10 years ago. So one day I was just doing a blast search with NPMA and found this. So all the way down on the screen, you can see that there's an E. coli genome with just a 40% identity with NPMA. And this genome was deposited from the UK as part of a big surveillance program, but was sort of just a sequence was deposited without specific annotation. So we thought, this might be a completely new enzyme. And we synthesized and expressed this new protein. And it turned out that this new enzyme, we now call NPMB, indeed conferred resistance to all aminoglycosides tested. And the overall tertiary structure was very similar to NPMA, but through X-ray crystallography, we showed that a structural change of a beta sheet linker was a requisite for target binding. So the way it works seemed a little bit different from NPMA, and sort of opening up a new venue of research into this group of enzymes. Finally, we are in the middle of this COVID-19 pandemic. And this pandemic has really accelerated sequencing capacity worldwide. And how can we leverage the capacity and infrastructure once the pandemic subsides? I think is a big challenge and opportunity for all of us in clinical microbiology. So in conclusions, high throughput sequencing of bacteria could transform how we practice infectious diseases. Molecular investigation of hospital epidemiology has largely been replaced by sequencing. Diagnosis, including identification and even susceptibility prediction, is currently the new frontier. Implementation remains a challenge moving forward. And finally, advances made in response to the COVID-19 pandemic could be potentially be leveraged to facilitate progress in this area. So with that, I'd like to end my talk. Thank you very much for your attention.
Thank you very much, Professor Doi, for the excellent presentation. Check, we don't have any question from the floor. Wait, I think we have a few more minutes. Can I just ask one or two questions? Since you mentioned about the the long turnaround time into genomic sequencing, what do you think is the ideal or acceptable turnaround time?
Pardon, could you repeat the last part?
Ideal or acceptable turnaround time for genomic sequencing in bacteriology.
Oh, turnaround time.
Yeah.
Yeah, I think there's still a large gap. So it has to be faster than what we do in clinical microbiology. That's an obvious one. So for species, it's probably should be the same day if possible, and susceptibility maybe at the same time or the day after, because the incremental costs need to be justified, and that is a function of turnaround time, I believe. And I don't think we're quite there yet.
Yeah. At the end of the day, we want to see the impact of this on our patients in terms of management and treatment. So you mentioned a lot of challenges that putting in genomic sequencing into incorporating it into our clinical practice. So what do you think is the best, or in what situation or indication is most impactful in your experience?
Right. So clearly, not limited to genome sequencing, but these the rapid diagnostics are deployed in blood cultures, more recently pneumonia. So conditions that are associated with worse outcome. At least that's been the case when I practice in the US. But I kept saying challenges. Sorry, but I think one big threshold is whether all this effort can go from add-on tests to a replacement of what we do. And as long as we're doing this sort of as an additional test, I think the indication will remain fairly narrow. Once we have methods, approaches that are comparable to what we do, which takes forever with culture, I think that would be a time for a breakthrough. Hopefully that'll be soon.
Yeah. Microbiology is still the traditional old way of doing things. Yep. So we hope that we can see a transformation in terms of putting in genomic sequencing as the test for microbiology. So thank you very much, Professor Doi, for the excellent presentation.
Thank you very much.